Hello to those joining us today. And in a couple of minutes, we are going to start. Meanwhile, if you want to write in the chat your name and where are you from, please do it. Okay, I think that we can start. So, good morning, good afternoon, or good evening, depending on your time zone. Welcome to this new webinar of the Trevor Platt Science Foundation. The foundation, as you know, is an international organization dedicated to capacity building in science, research, and education related to our oceans, working towards stewardship of our planet for future generations. Today's webinar is one of the capacity building activities of the foundation. And before introducing our speaker, just a couple of issues. You notice your microphones are off, and so please write your questions in the Q&A session, and we'll get to them after the talk. So today I have the pleasure to introduce our speaker, Dr. Victor Martinez Vicente. Victor is a senior bio-optical oceanographer at Plymouth Marine Laboratory, specializing in satellite oceancolor remote sensing of marine ecosystems. With over 20 years experience, his research advances the monitoring of marine biodiversity, carbon cycling, and plastic pollution through innovative multi-mission Earth observation approaches and international collaboration. So Victor will talk about advancing satellite-based monitoring of marine carbon pools, phytoplankton diversity, and marine litter pollution. So Victor, the floor is yours.
Okay. Let me see if I can share this one. Okay. So can you see the slides?
Yes.
Yeah. Okay, fantastic. Well, good morning, good afternoon, good evening. We never know which part of the world we are. As Vivian said, my name is Victor, and I'm very, very pleased to be here today. I don't share the office, but my office is very close to that of Shuba, and for a time it was very close to the one of Trevor as well, here at Plymouth. so at the Plymouth Marine Laboratory. So it is a great honor to be invited in this event, which is part of a capacity building from this fantastic initiative. So I have been at Plymouth for over 20 years. I think next year it will be 25. And I started here as a junior marine bio-optic in situ scientist going to ships and sampling. And I have derived into larger scope problems, including satellite algorithm development, but always very linked to the connection to the in situ data. So my talk is going to be most of all a call for not thinking that the satellites are a silver bullet, but that they are more powerful or most powerful when they are combined with in situ and other kinds of data like models. So here we go. I hope that you will enjoy. And of course, if you have any questions, put them in the chat or in the question and answer thing. I cannot see it, so someone will have to tell me what to do with that. So we have a large problem at the moment. We have the combination of three crises that are hitting humanity more or less in the same time, and is if like me, you have children, or you don't have children but you worry about the future, these problems, these threats are really something that are really close in time, and where we need to have some actionable activities. So one of them is the global change, as you know, or you may not know, but surely know now. There is an increase in global temperatures. We have been experiencing them in terms of heat waves being more common here in the UK, but there is an increase in global temperatures, which is predicted to increase over the next 70 years. There is this climate change is affecting land and marine environments, and the marine ecosystem have a big role to play in it. At the same time, we have detected a decay in the biodiversity, so in the numbers of species or biological species going forward, and they are not predicted to increase if we don't do anything. So the trend is a decrease of the biodiversity. Simultaneously, the human activities are increasing because the population is increasing, and one reflection of this is the increase in the use of marine, in the use of debris, or in the use of plastic commodity products in the day-to-day life, and ultimately a lot of them end up in the ocean. Again, the trend is not one of decrease. The projections that we had 10 years ago were of an increase. I don't know if there has been any newest update to these trends that I show in the last figure here, but giving the current levels of increasing population, I expect that these trends will continue as they are, because I haven't heard of any radical action being taken. So in this context of increasing temperatures, reduction of biodiversity, and increase of human impacts due to marine litter, there is a need to monitor what is the speed of this change and whether when we do some implementation of policies to reduce or tackle some of them, there are some effects. Therefore, there is a need to monitor these three aspects of change, especially in the marine ecosystem. The marine ecosystem is very important because in terms of climate change, they have one of the most active and largest pools of carbon in the ocean, in the world, and diversity, in particular the diversity of phytoplankton, controls the ecosystem function, and it is the base of all the ecosystems and biogeochemical functions, but it is difficult to monitor indeed. And then the marine litter pollution is very widespread. It is patchy, but it requires some synoptic monitoring. With the capabilities that we have, in situ data are limited because they don't provide repeated observations regularly in many places. They tend to have time series. They also tend to have not be harmonized in terms of the measurements, how they measure chlorophyll, which is an indicator of phytoplankton biomass, in some areas may differ to the other, and even within methods, within different laboratories using same methods, there can be substantial differences. There is, however, therefore, there is the need of resource to additional tools for monitoring, such as remote sensing from satellites, and in particular, one of the physical technologies that are useful to monitor phytoplankton, the carbon, and potentially the floating litter. It is the optical remote sensing. However, the challenge is to translate these optical signals into some meaningful variables, and that is mostly the topic of my research. So what I have been doing, or what I have been leaning towards, is to develop Earth observation using combinations of in situ, satellite data, and model data to improve the estimates in carbon, to benefit the climate change, to improve the monitoring of diversity of phytoplankton, to be able to monitor better the biodiversity decline, and to find ways to explore the ways to find measures of marine litter, which is from a point of view of human pollution. So a number of projects, in particular from the European Space Agency, have been supporting this effort in the last years. And here is a list of them. Within them, there is a number of collaborators and institutes that have with whom I have been working, and there are really too many to cite in there. So I invite you to go to each of the websites for each of these projects and find the actual people and the actual institutions that have been helping myself and Plymouth Marine Laboratory to deliver on all these projects. I am sorry not having had the time to put them all in there, but they are really a lot. And I think an important message is the need for interdisciplinary research when tackling these problems. So now I'm going to go in this talk a little bit one by one through these problems, starting with the carbon. To monitor carbon from space, you have to we rely on the reflectance, which is essentially the watercolor, and it is spread among a number of wavelengths. We we are able then to retrieve. This is a very nice graph from Dave Siegel, where you have starting from the top of the atmosphere, you transform it into remote sensing reflectance, and that can give you estimates, indirect estimate of carbon proxy, like chlorophyll, which has been used in the past and is the most standard oceancolour product, as well as other products like the particulate organic carbon, the inherent optical properties, and from that, and in combination with other variables, you can get to phytoplankton carbon estimates and phytoplankton size classes, PSS, and also phytoplankton community composition. All of those can feed into NPP, which is the primary production, that can be then connected to other types of data from, or used in other data, in other context for validating or comparing to other models. And from the combination with the models, you can eventually get to carbon export, which is really a very important ecosystem function for the marine ecosystem. Now, one of the big gaps in the research is coastal carbon. And of all of the variables that I presented in the present slide, in coastal carbon, the problem from remote sensing is to try to separate the different composition, the different components that affect the color of the water. So we were awarded a contract with the European Space Agency to do just that, to look at carbon components in coastal waters, which really contribute disproportionately to the primary production and act as an interface of a carbon exchange between the land and the ocean. So we look around the European areas, as you can see. We look at the areas that connect the continental shelf to the open ocean via upwelling, and we selected an area of very active upwelling in the north of Spain as a case study, and also two plumes between at the edge of the plume and the coast and the continental shelf, and we selected some areas in the Baltic Sea. To do so, then we have been doing some algorithm testing, and we have been comparing in situ observations for dissolved organic carbon, and test some different algorithms, also for particulate organic carbon, and test some algorithms, and also for primary production, and test some refinement in terms of chlorophyll detection, to come to a harmonized coastal carbon from satellite. This we are expecting to produce 10 years of carbon products. These carbon products in 300-meter resolutions, and some experimental data at 60-meter resolution. This project is still ongoing and will finish next year. So keep your radar on for the products if they may be useful for you. The other part of the carbon jigsaw here, especially in the coastal areas, is what can we do to prepare for the new satellites coming in line? And these satellites that are coming online have much higher capabilities. They are higher spectral resolution, and they have a higher spatial resolution, in particular CHIME. And LSTM is looking at the high spatial resolution of the sea surface temperature. So we have to be prepared for these satellites. They will be launched after around 2029, 2030. And to do so, we have designed an in situ sampling campaign, which will allow us to develop new algorithms, so that when the satellites come, in fact, come up to space and begin deliver data, we're already ready to work on the data and deliver measurements, possibly to modelers and so on. So The in situ campaigns that are taking place almost as we speak, we have more sampling next week, are centered in around the Western English Channel. This is Plymouth here, and there is some sampling that goes up to the estuary of Tamar, and then the other one is in the north of Italy, around Venice, where we'll be looking at perpendicular transects and collecting a data set that will look to be very, very complete in terms of hyperspectral radiometry and carbon products. But this is a technique that we need to be collecting this in situ data, not only for the carbon. We need to be doing this if we want to have any advance into the future to do new phytoplankton diversity algorithms. The phytoplankton diversity is at the base of the biogeochemical and ecosystem functions, as I said before, and these hyperspectral data allow to separate the different phytoplankton functional types, especially when they are linked to their absorption peaks. Now, the challenge, again, I don't like to collect or to do the easy things. People, so always looking for the most difficult bit of work, and therefore we're looking at the coastal areas in this case, again, where there are other components that affect the optical response, like the reflectance or the color of the water. So in coastal waters, there is a point where there is so much in the water that you're not able to see phytoplankton groups or even the chlorophyll itself. But we're trying to see up to what extent, at what point we can push and take advantage of hyperspectral and where things completely break down and where things are possibly salvageable. And to do so, we collected with other collaborators, CNR in Italy, Laboratoire de Villefranche, University of Maine. We equipped and together with a European project ongoing, the TREC mission, we installed optical instrumentation and collected additional data on a sailing vessel, the Tara Oceans, the Tara. And during 2023 and 2024, we collected a series of 200 stations, and we collected over 600 samples and collected thousands of continuous hyperspectral measurements. And we demonstrated that the quality of the hyperspectral radiance on sailing vessels collected with an autonomous platform called SORAD, it is of sufficient quality to be used for algorithm development and satellite validation. So opening new avenues to what can be done in situ and how can we move forward to collect more in situ data, which is critical for the development of algorithms. So an example of the data we collected is here, courtesy of my colleague Tom Jordan, here at PML. And it marks the positions of the matchups that we got with different satellites and the shape of the reflectances with calculated uncertainties following the most up-to-date techniques. These data are being worked up and being prepared to publish. My colleague Vittorio Brando from CNR Italy is condensing or collating this in the form of a data paper that will be available to the community really very soon. So again, keep your eyes open to this and check the website or our social media to see that when this paper is published. I'm sure it will be a very interesting dataset for the community. So in terms of more into the biodiversity, it is good to collect in situ data. It is good to test algorithms, but what it is best is to connect the algorithms to real satellite data and to give an application to policy. So in this context, we looked at another project called Phytodiverse in the areas in the North Atlantic, the North Adriatic, and the Arctic regions, where we were trying to deploy algorithms for phytoplankton diversity based on hyperspectral data to compute indicators such as trends that can be used in policymaking and monitoring that decrease of or changes in phytoplankton diversity. So the first thing we're doing, this is a project that started only last year, it lasts 3 years. First thing we're doing is a comparison of algorithms. We collated a number of in situ data. We combined the in situ pigments and in situ radiometry. My colleague Tom Jordan leading on that effort. And will be delivering. We have had just the first round of comparison of algorithms, but this is a open call to participation for if you have some interest and some in situ data that you want to participate in this initiative, please let me know. And then the final bit of the biodiversity is a project called Plantoscope. And with it, it is even looking for even more into the future, where we have new algorithms that are able to connect satellites to eDNA or molecular biology. But they do need a lot of in situ data that currently do not exist. So to improve that, we have engaged with citizen sailors and distributed. You see all these points are sampling points from citizen sciences who have taken a kit on their sailing boat, and they have followed a strict protocol to provide a data collection. But this data collection is really not a blind one, because each of these samples is one costly material and effort to be taken. So we at PML, in collaboration with our partners, and financed by the European Space Agent, we have designed an app that allows the computation of satellite overpass wherever you are, and gives you some indication of how good or how bad the match with potential in situ data can be, so that you can really go into the field and in advance know whether it is worth or not to do the effort on collecting the sample. So you can search it up. It is at the moment available, I think, in wherever you get your apps. I don't want to say commercial names, but if you want to access them, please, and you cannot find this Plankton Space app, please let me know via email or through LinkedIn. Now, the other problem that we have is marine pollution, and this is a nice slide produced by Professor Chuan-Min Hu from the University of South Florida, where he summarized a large amount of floating material that can be confused with marine litter. Marine litter pollution, it is, as I said, a big problem for the ocean, and it is again a challenge for optical remote sensing, because you have all these materials that also produce a high reflectance in a similar shape as marine litter does. So I have been working with a task force on marine litter from the IOCCG, and we have produced a a paper where we try to harmonize. The first thing we need to do is to try to there is very little or relatively little research on this field, and we try to harmonize the language. And this is what we did with my colleague Loneke, Godin Murphy, in a recent paper where we try to make that everyone use a similar terminology. So please, if you're looking to work into remote sensing of marine litter, you please have a look at this paper and don't try to reinvent the wheel, because it is just very useful to keep a common language for advancing the field. And the other thing I want to say is just, I'm keep on saying publicity, I'm sorry, is that we have a task force on remote sensing in IOCCG, and we're looking for people who would like to participate into this initiative. So if you're interested, again, contact me, and we'll see if we can find you a place to give a hand. So what I did in a couple of other projects, in particular one called Frontal, was with the support from Peter Miller, our colleague here, who's leading the effort on this work, is to combine satellite fields from sea surface temperature, or maybe potentially from oceancolour, and detect some fronts with a special algorithm, and also through model fields predict where the particles will be moving to, and see when they do not match, to provide a front risk index. This is so because we realize that algorithms that there are not enough in situ data to develop robust enough algorithms for marine litter. So we decided that at the very least we should be able to indicate where there is potential for litter accumulation, and where potentially sampling could be conducted to see more validation data for the satellite. So it is always this combination of different data. So here is an example of a simulation at a larger scale of mass concentration of litter from a numerical model at a larger scale, and then at a shorter scale close to the area of Da Nang in Vietnam, where the project was taking place. So when we see from the models where the particles tend to accumulate, and we combine that with where we see where the fronts are, we can get two maps similar to these, which show the risk index of combining the in situ, the satellite frontal areas, and where the models say there are accumulations of litters. So here you can see that the model is predicting in several areas, but on the fronts we see that some of the areas for fronts allow have a greater risk than other of accumulation. This changes with season. Obviously, you have the monsoon there, but we are still working on these results, and hopefully a publication will come up soon. So now I realize I have been talking perhaps for too long, and I apologize for this, but I'm happy to reach to some conclusions here. I hope that I have convinced you that there are some aspects of the climate change, biodiversity loss, and marine pollution that can be monitored from satellite, but they require the development of new tools that can match the new capabilities from the new satellites coming online. In particular, challenging areas are the blind spots, or not blind spot, but challenging areas are coastal and near shore waters, And my main point would be that, as I said at the beginning, the satellites are not a silver bullet on their own. I think it is important that we keep in mind that where they gain the major power is by using them in combination with in situ observations and with modeled data, and or with modeled data, whatever you can use. And I think with that, I finish. Again, I thank you for the time today for inviting me and to give this seminar. And also a big thank you to our sponsors, the European Space Agency majorly, and all the partners in the different teams for all the different projects, which again, I'm sorry I haven't put everyone in the slides. I usually do, but there's really too many. So I hope that you can go into the websites and learn more of these projects into the future. So thank you very much.
Thank you, Victor, for a very interesting webinar. Really, so many new things and so exciting. All the tools combining in situ, satellite modeling, and I guess now AI should be somewhere there. So I will check the Q&A part. Okay, we are a question from Sarat Tripathi and asked, how transferable is the algorithm developed for UK and Italian coastal waters to other turbid coastal environments, including Arctic fjords?
Okay, can you say that? I cannot see the questions. Can I read it, or is it only to you?
Let me see. I will see if I can transfer it to the chat. You can see the chat, isn't it?
Yes.
Okay, I will put it in the chat. Let me see if it allows me to do
I've
that.
shared it to Victor.
Okay, good. Thank you. Thank you, Patricia.
Thank you very much. Yes, great question. It's this is the problem. So we are trying to collect and make our algorithms as transferable as we can. We will produce our algorithms with some estimate of their uncertainty. So when we transfer to a different area, we will know at least their uncertainty. And indeed, if you have in situ data to compare, that is what should be done with any coastal algorithm. But we're trying to map some of the products that we are computing using optical water types that you could find in a way geographically independent. So we are hoping that there will be some transferability, but indeed transferring whichever algorithm from a coastal area to another coastal area, it is always a challenge. So the best that you can do is to see what is the uncertainty in the products in the local area with in situ data. So if you have in situ data, then transferability is easier. But thank you for the question. Yeah.
Thank you, Victor. Yes, I agree with you. I'm also an in situ person. I value all the tools, but it's always good to have time to time a check with reality, the real world. There is another question from Pratiksha Desmond from Goa. And the first, she has two questions, sorry. Is it possible to detect EPS produced by phytoplankton from satellite? That's the first.
Is it possible to produce what? T?
To detect EPS. I'm not sure what. I thought about. I guess so.
I think this is this sort of aggregates that the phytoplankton produce. We haven't done. This is a very interesting topic, but we haven't really addressed this in these projects. The algorithms that we're working on are mostly for the coastal area, so our problem mainly is the interference from uh, minerals on whatever signal we have. For the open ocean, different parts of the carbon pools are as yet, I think, unexplored. I don't know if anyone has. But I wonder, in terms of TPS, what is their nominal sizes? Bear in mind that the most visible parts in terms of sizes, are the smaller cells, which are coming in greater numbers. And I don't know what is the abundance in terms of number of particles and their size for this TPS, but it is indeed a very good question. And I don't know also what depth they form. So yes, it is. We haven't been looking at that. Very exciting question. I have no answer for it. Sorry.
Yeah, good. Thanks. Actually, on that subject, just a brief, I don't know, comment or uncertainty. Well, if Pratisha can maybe tell us more about what is meant by EPS. I sort of recall that has to do also with thickness, thickness of the different particle sizes and bacteria and so on. So it's more than just a particle, but it's an agent that helps to nucleate others. If I'm not mistaken, that's what I recall from some years ago.
Again,
So
not my field
yeah,
either, but very interesting.
yeah. So that is interesting. And the second question is, how does the satellite differentiate POC from phytoplankton carbon? So total POC from phytoplankton carbon.
Sorry, what is the question?
How does satellite differentiate particulate organic carbon from phytoplankton carbon specifically?
Yes, we in the projects that I'm working on, I presented today, we're not looking at that distinction. It is indeed a big question, and it is related a little bit to the previous question, right? The particulate organic carbon pool, it's composed of living and not living. I think the problem there it is that the phytoplankton carbon, it is still from space. It's still not very well developed. I did some work on the phytoplankton carbon 15 years ago, and came to the conclusion that it has got very large uncertainties. So I think it is. Uh, eh, eh, as yet, I don't think that we're there to be giving a strong or very good indications of that. However, we have a couple of projects working at Plymouth Marine Laboratory, and my colleague Hema Kulk is working on that at the moment, intercomparing and testing different algorithms. But then again, it depends a little bit on the in situ data and how standardized is the computation of phytoplankton carbon in the literature. Many ways, many parameters, carbon to size changes, very different things. So yes, interesting question. Not my current topic of research, but indeed a matter to be discussed further.
Yeah, a good field to develop.
Yes.
So thank you. Another question from Ratna Suhita. Hi, Victor. Thanks for the great presentation. The app you mentioned caught my attention. I downloaded the app and I saw the item score while looking at the satellite path planner. What does score mean here?
That was very quick. I think you didn't listen to the rest of the presentation, but it is good to. Yes, the score is a combination of different metrics, the weather forecast, the cloud coverage, I think it's got the wind. You can check it out in the app itself if you click on the matching overpass. You can click that it is where it is. It has got full detail on what make up the actual index. So have a look. If you click on I think if you click in the actual predicted matchup on the score, it will explain to you what are the components of that. But I'm very happy that it works like that, and that you're already testing it. So yes, please do send us feedback on the app. I think in the app itself, you can give feedback to the algorithm, to the app developer. So please do feel free to send messages and suggestions also to improve.
So great, you got buyers as directly from your webinar. So another question from Milton Campbell. Say hi, Victor. Milton Campbell from INPE, Brazil. Thank you for the interesting presentation. I have a question regarding hyperspectral data. Could you comment on the signal-to-noise ratio of current high-resolution hyperspectral satellite missions, such as ENMAP and PRISMA, particularly in comparison with PACE?
Hi, Milton. Nice that you're attending this seminar. In particular, we don't use Nmap and Prisma because they don't fulfill the requirements for the projects that we lead. So I haven't yet worked with Nmap or Prisma in any of the research projects that I have. I know that PACE has a very good signal to noise ratio because it is tuned to ocean radiometer. So, and also because of the 1 kilometer footprint, so it has a greater. It should have a greater signal to noise. But again, I am not too familiar with Nmap and Prisma. We attempted to use them at the beginning of some of the projects, but access and targeting, you had to target the satellite in some occasions, for particular to Prisma. It was really challenging. Now that we have an operational satellite, we have decided to use PACE for our research, and we will expect that we will be using CHIME, which is what we will come for which we are preparing now for the next 3 years.
Thank you. Another question from Ravi Naik. Thanks, Victor, for your interesting talk. It is regarding marine plastic pollution. How effective is remote sensing tool to detect the microplastics in open ocean?
Okay, well, I did some research. I didn't manage to publish it at the time, but there are some people who have done since much better research on this field. I'll put see if I can put the reference. I think they Coestna et al. 2025, something like that, or 24. I'm not sure. But Daniel Kostner published a paper in limnology and oceanography. He did a similar experiment to what I did, but much better with a great amount of laboratory resources. And he measured the optical properties of microplastics. He came, I think, to the same conclusion that I did, but he published it. So he has something in the literature in which indeed they for the concentrations of microplastics to be seen from space, they would need to be very high, or they would need to have a high imaginary index of refraction, so they need to be very colorful. However, the majority of microplastics in the ocean tend to be not tend to be white, and as far as I know. So and depending on the yes, and they're not insufficient numbers, I would say, to affect the reflectance with the satellites that we have at the moment. That is, if you look at the visible part of the spectra only. If you look at the shortwave infrared, it would be even harder because a lot of the signal in the shortwave infrared is absorbed by the water. The the microplastics will have a problem with that in that they are in the water, immersed in the water. So any signal in the shortwave infrared will be absorbed by the water itself. So I think for what I know now, but maybe these things will change. But I have quite a confidence that direct measurement of microplastics, and this is the larger microplastic, it is not really possible for the moment with the technology that we have. Yeah, it's a good question. It is important to keep on thinking about it and see, and hopefully we will never get to a concentration where we can detect it from space, because that would mean that we are in a soup of plastic.
Yes, actually, I was thinking the same. It's quite tricky. We know there are a lot of microplastics in the ocean, but they are difficult to detect. But I was also thinking, hopefully, there will never be enough that we could detect them. Yeah. Another question from Gunalan Tulikambi. Can marine litter windrows be tracked using remote sensing? And can the polymer types within those windrows be detected or classified?
Well, this is a good question. First, I am not totally in favor of calling the floating accumulations windrows, because windrows can be accumulated by, I mean, the floating material can be accumulated by tidal currents as well. So it's not only a matter of wind accumulating them. So this is something that it is in the literature, but is again one of the terminologies that we perhaps should be aligned somehow. I don't know how, but this is something. In terms of whether they can be tracked using remote sensing, I definitely think yes, depending on the relative size between the pixel, the ground sampling distance of the sensor, and the the the, the row of floating litter itself, the wind row itself. If they are below the pixel resolution, then they can't. Now, there is another problem with the windrows, is their permanence. If they tend to be very ephemeral, so they last very little on the water. So you have to be pointing with a satellite at the time where there is a windrow there. In the open ocean, maybe there is a chance to be finding them. In coastal and tidal environments, in tidal environments, there may be also some more permanent features, but as you go out of tidal into coastal waters, they're really very, very changeable. We've tried to match different satellites pointing at the same place, and in matter of minutes, things can change. So it's very difficult. In terms of the polymers, for the moment, no, but I think Heidi. I think Heidi Diersen just published a paper with her student on some sort of floating litter measurements, and I think they found a way, or on some wavelengths which are particular, and there could be a chance. Heidi Diersen. Yes, and and there could be a chance to have a go at separating, but the matrix of the mixed. This is something that I put in the slide. The mix matrix can be very challenging, not only because it's mixed, but also because you have biofouling, masking, and you have a degradation of the plastic. In fin, I think at the moment we are in the position to detect floating matter aggregations, either windrows or other, and then from that have a basis to direct maybe other efforts, either from drones or from directed in situ sampling to sort of support other types of sampling rather than to sample on it directly. I think that could be a role while technology advances and we have a different technology that can help with this.
Okay, thank you. A question from Radna Suhita. As far as I'm concerned, theoretically, primary production and new primary production are two different things. How do we differentiate these two when it comes to data processing and results interpretation?
The first part of primary production and what was the other?
And NPP, new primary production.
Oh, wow. No, we haven't looked into new primary production and net primary production. Is that that's the question?
Well, actually, it says PP and NPP. I'm not sure if NPP it refers to net primary production.
Net primary production. I think it is a question referred to the total gross primary production and net primary production. We're not, yes, we're using a published algorithm on net primary production. Yes, it is based on the Morel algorithm on the 91 and then implemented at PML by Smythe et al. In 2005. It's a wavelength resolving algorithm. I think it has input from sea surface temperature, but we are looking at it in coastal waters and we are trying to validate it with some in situ observations of net primary production. So we're hoping we're looking at net primary production. I don't know. I'm not really an expert myself on primary production, so I cannot really go very deep into this question.
It's another complex issue. It's like phytoplankton diversity and TP. They are not the main variable, but rather they are more complex phytoplankton properties. So. And finally, there is a question from Ana Dogliotti, my colleague here from Buenos Aires. And she says, does CHIME, that's CHIME, has the signal to noise ratio required for oceancolour?
Well, we hope, Anna, hello, how are you doing? Yes, well, we hope it does because we're going to be using it for marine application and especially for coastal application. I think at least for coastal, even if it doesn't have a really very good signal to noise, there will be sufficient suspended matter and sufficient signal for it to capture things. Anyway, we're talking about 30-meter resolution. So using 30-meter resolution in open ocean, it's a bit of an overkill. So I think would be we don't know. I don't know if it will be sufficient for oceancolour, but I think it is good enough. I will double check, but we have based a whole project into it, so it better will.
Okay, these are the questions that I see on the Q&A. So we had a very big and active audience today. So thank you very much, Victor, for your interesting talk. It was very active with a lot of issues. And thank you very much to all the people that joined the audience, and also especially to the Earth Center for hosting the webinars. And from next week or from next month, we are not going to continue with this regular TPSF general webinars, because we are starting next week the series of lectures previous to the training course that will go together with the symposium in India. Our annual meeting that's going to be held in between Hyderabad, the course, and Kochi in the south of India. So, we hope that apart from the students that, of course, are going to be attending these lectures, all the members and all colleagues, please join us also. From next Tuesday, you're going to get the announcements, and we hope to see you there. So thanks again, Victor and everybody. Patricia.
Okay, thank you very much.
Okay.
Thank you for inviting me. Thanks.
Okay, bye-bye.
Good luck. Thank you.
Bye.
Okay.
Okay. Bye-bye. Bye, Patricia.
Bye.
Thank you.